All right, maybe I get started. Good morning, everyone. Today we have Professor Shiladitya Sengupta. He is a co-director of the Center of Engineered Therapeutics at the Brigham and Women's Hospital, who is also an associate professor of medicine at Harvard Medical School. His laboratory uses tools from nanotechnology to study biology at scales not achievable using classical approaches, commercially use the understanding of biology and medical problems to design next generation of nanomedicine. Dr. Sengupta trained at the All India Institute of Medical Sciences, received his PhD in pharmacology, is a Nehru Scholar and Chevening Scholar from the University of Cambridge, UK, and completed his fellowship in biological engineering at MIT. He developed the first layer-by-layer nanoparticle, Nature 2005, and recently discovered a novel mechanism of immune evasion by cancer cells via nanotube-mediated mitochondrial hijacking from immune cells, Nature Nano 2022. Many of his inventions and research, 51 granting and pending patents in US alone, have been commercialized or led to startups, including Cerulinian Pharma, Biome Therapeutics, Farcas Biosciences, Elysium Therapeutics, which have raised over 300 million in R&D funding, or have been commercialized, Nimulid, Canstrip, etc. And some of them are in phase 3 trials, and some others are phase 1 trials. He received the Era Hope Scholar Award from US DoD, the TR35 Innovator Award, among many other awards. The floor is yours, Shiladit.
Thank you, Mehmet. And is it clear?
Yes.
Perfect. Yeah. So thank you for the kind introduction and also for the invitation to give this talk. And I can see Ali has also joined in. So hi, Ali.
Hi, Shiladit. Great to have you here. Thank you very much.
Yes. So these are my disclosures. I co-founded these companies and I have equity in these. So my lab basically is at the interface of engineering and medicine at the Brigham and Women's Hospital. And the work that I'm going to discuss today about, how cancer cells escape or treatment and grow. I'm going to touch upon the works of outstanding fellows and graduate students in the lab. Some of them are listed here. And I've been very, very fortunate to have these, fine scientists to work in my lab. And we have a completely diverse, set of people. And if you're interested in looking at us as a potential place to join, happy to basically have a chat. So what does my lab do? What we try to do is to apply tools from nanotechnology and bioengineering to basically explore two aspects. First, on the basic science front, the fundamental aspects of biology. The principal question that we are asking is, how do tumor cells and stroma communicate? Okay, and that information, the fundamental knowledge that we gain is then basically applied to develop new treatments and translational platforms. So there's a huge intersection between basic science and applied science in my laboratory. At the same time, there are also projects where we start off with an application and then we go back and find the basic science side of it, the fundamental side of it. And the connecting link is basically our understanding of nanotechnology and bioengineering. So many of you have probably seen this video of a T cell attacking a cancer cell. And this is where cancer therapeutics is heading. But the way I like to kind of, look at science is how it has evolved over time, over the history of it. It's absolutely fascinating. In fact, cancer is actually a very old disease. So the earliest description of cancers pretty much come in the Edwin and Ebers papyrus, which is about 2,500 BC. And there's a field of science known as paleopathology, where they study, old mummies, for example. And this is a scan of this mummy, and they actually found metastatic lesions on the bone. The word cancer was actually coined by Hippocrates. He called it carcinos. It comes from the word crab-like. Now, I am a pharmacologist. I love developing drugs. And so as a result, for me, basically, to be able to trace, to be able to develop new drugs, it's very important to trace where we came from, how we started. And there's a very interesting story about how the first chemotherapy came about. So what happened during World War II was that this ship, this is John Harvey, had docked at a small Italian town, Bari, and it got bombed. And it sank, and people around that village basically died. So US Navy basically investigated as to what caused the people to die. And it turned out that US John Harvey was carrying mustard gas, and this is the structure of mustard gas. And when the medical examiner basically investigated people who died, he found that there were almost zero blood count. So the blood cells had actually been wiped out in those patients who had died. And that then kind of led to Goodman and Gilman, who were researching derivatives of mustard gas. So this is the first drug that was actually developed as a potential treatment for blood cancer. And this is the first paper that was published in Science that initiated the field of cancer chemotherapy. So from there, we have now reached a stage where we can manipulate the immune system to potentially cure cancers. So what do we do? We basically ask questions at three different aspects of how a cancer grows, and it addresses the question, how does cancer escape? And what do I mean by that? In the sense that we have so many treatments, why is it that the tumor still basically manages to grow? So at a fundamental level, we are basically asking these questions: how do cells communicate with each other? And especially at the nanoscale level. The second aspect, and the reason why people, why people die of cancer is because the treatment fails. So why is it that, how is it that the tumor is able to survive chemotherapy or treatment? So why is it that therapy fails? And we are asking that question. The third aspect, which is fascinating, is the fact that the cancer cells have evolved mechanisms to avoid the immune system. And how does it do that? So the current concept was that, how does it expresses immune checkpoints, and it can escape T-cell immunotherapy. But what are the newer concepts that are emerging? And these are the three themes that I would touch upon in my talk today. So I got into cancer because I got very interested in the formation of blood vessels. And this was a long time back when I was doing a PhD at Cambridge University. The field of angiogenesis was started by Judah Folkman, who was Bob Langer's mentor. And what he basically had proposed was, as the tumor is growing, it needs blood vessels, and can you shut it down? And this picture basically kind of captures, how a tumor cross-section looks like. So, for example, these are the cancer cells. This you can actually see is a blood vessel. These are the RBCs in the blood. And what is interesting is this huge amount of matrix that surrounds the tissue. And I got very excited about studying both the matrix as well as what are the drivers that actually cause these blood vessels to form. And I worked on a novel growth factor at that point of time called HGF-SF, or hepatocyte growth factor, and how it actually promotes blood vessels formation. Now, what is interesting is basically the concept of shutting down these blood vessels by inhibiting factors like HGF or VEGF was at that point of time very hot. But there were these papers that started coming out which talked about if you actually shut down the blood vessels, you can actually promote metastasis. Okay. The question is, how do you kill those cells which would have metastasized even after blood vessels are shut down? And this was a question that I was actually thinking about while at MIT. And I came up with this very simple concept. And this is the first, layer by layer nanoparticle that we had designed for cancer chemotherapy. And this video that I'm going to show you it's a cartoon video, which captures the concept of what we did. So as I said, as a tumor starts growing, and this happens because there is a mutation and the cancer cells are proliferating rapidly. So what happens is that when it reaches a certain volume, let's say about 2 millimeter cube in volume, the core of the tumor basically starts dying because it has no supply of oxygen and nutrients. And as it starts dying, it creates a gradient of growth factors, which then basically recruits blood vessels from the nearest larger blood supply. This is a process known as angiogenesis. This is what Judah Folkman had suggested that we should actually shut down. So the goal is, if we can actually shut down these new blood vessels that are formed, or the process of angiogenesis, you can also potentially inhibit metastasis, because these are the blood vessels through which the cancer cells metastasize to distant tissues. So this is the concept of anti-angiogenesis therapy. You give a drug, you shrink the blood vessels, but now what happens is the tumor basically is very large and the blood supply is shut down. The core of the tumor again becomes hypoxic, and it's the hypoxia and the enriched growth factors that basically keeps the tumor alive. So then the concept is, can you give a chemotherapy agent? The problem is, how do you deliver chemotherapy when blood supply is cut off? And that's the reason why relapse is happening. So what we came up with is a very simple concept of a balloon inside a balloon, and reduced it to the nanoscale. So we had this big balloon and a small balloon inside. In the bigger balloon, in the outer layer, we had an anti-angiogenesis drug. And the inside balloon, the smaller layer, we had a chemotherapeutic agent. And when you reduce it to nanoscale and you inject it IV, it keeps circulating and it starts accumulating in the leaky blood vessels of the tumor. So it starts coming out into the tumor, okay? And it accumulates there. And there, the first balloon, the outer layer bursts, releasing the anti-angiogenic agent. And that basically then shrinks the blood supply. Okay, so now you have kind of, these smaller balloons, the balloons inside are now stuck in the tumor. It can't escape. And the tumor has become hypoxic. We called it the nanocell. The drug then, the inner balloon is loaded up with an anti-cancer drug, which is then triggered by enzymes that are being released by the dying cancer cells. So these entrapped drugs start coming out. This is the chemotherapy agent that comes out. And so what we have been able to achieve by layer synthesis of these nanoparticles were sequential release of these two drugs. And the second drug then comes out and kills those cells which would have otherwise survived and shrinks the tumor. And if you look at the blood supply, this is the reason. So this is the blood supply to a normal tissue. You can see everything is very nicely networked. Everything is connected with each other. This is the blood supply to a tumor. You can see open ends here, for example. Okay. So it's completely distinct. And you can actually leverage this difference to deliver nanoparticles to tumors. So this paper was published in Nature, and this got translated into a company called Cerulean. And in fact, this patient actually got treated with this drug and his tumor shrank, and CNN did a very nice story around this. But what happens is, despite all advancements, patients still die. And why is it? So the way I look at it is, we have a very reactive way of actually treating cancer. So imagine, this is a tumor, this is a drug, okay? And, you're almost responding that the tumor is actually shrinking. Here in this case, imagine the car is being dragged out of water, okay? And this is success in terms of chemotherapy outcomes. You're almost there. The tumor is shrinking. And then the drug stops working. It crashes. And then the tumor basically starts expanding again. So what you do is you come up with a more powerful drug. And again, you are winning the war against the tumor till it crashes. So, and you go through cycles of these. And at some point of time, you exhaust all treatments. What do you do then? So I think it's very important to go back and start thinking about why is it that the tumor basically is relapsing? Why is it that the tumor can escape all these powerful therapeutics that we're developing? And the important question that comes up, a point that comes up is the fact that we don't actually have good models to predict, outcomes. So for example, these are reports that you can get from, say, for example, foundation medicine. Okay, these are in a patient who's diagnosed today with tumor will actually get a genetic analysis done, and based on the genome analysis, you would see that this patient has a MET amplification, will potentially get a cabozantinib, will respond to cabozantinib. There is also an AKT2 amplification, and the patient can potentially respond to everolimus and drugs of that class. There was a KRAS mutation, so this patient will also respond to trametinib. Okay, and then again, there are patients who basically don't have some mutations, but there are no drugs that can potentially be used for these patients. So how do you basically, so this poses questions, how do you first select which drug to use? Do you give this drug or this drug or this drug? You don't really know whether this patient will respond. So this is and even if there is a mutation present and the patient is supposed to respond, very often it doesn't happen. So for example, in this study, you can actually see that this 61 patients out of 146 were had mutated KRAS. And of these 8 patients were treated with a drug called cetuximab. Okay, and these patients were not supposed to respond. Okay, so none of them actually responded, which was fine. But anybody who has a wild-type KRAS protein will get cetuximab as the drug of choice. So there were 60 such patients who got cetuximab. They had the target present based on genetic analysis. But it seems that 63% of those patients failed to respond, even when the target was present. So how do you really select the right patient? And we don't really have tools to do that. So what is the barrier? Okay, so why is it that genetic analysis alone is not good enough? And this stems from the fact that in the tumor, there's a lot of heterogeneity. So this cancer cell, for example, is genetically different from this cancer cell than this, and it keeps evolving as the tumor basically mutates very fast. So how do you basically then kind of design what treatment to use? So that was the genetic heterogeneity at a cellular level. What we observed is basically even the tumor stroma is heterogeneous. So for example, this is head and neck cancer patients, and a low-grade tumor versus a high-grade tumor, you can actually see that between the 2 grades of tumors in these patients itself, there's a difference between the composition of the extracellular matrix. And so we actually kind of looked at different patients. There are only 6 proteins which are actually conserved between all patients, between a low grade, and 7 patients which had 7 proteins which are conserved between patients in a high grade tumor. Similarly, in colorectal cancer, again, high grade and low grades, completely different profiles. And there are certain proteins which are actually conserved. So what we did was we started thinking about, could we start, developing a platform where we can actually test for drugs which would work in a patient exactly like the way you prescribe antibiotics. And so the way you prescribe antibiotics to a patient is, when you have a sore throat, you take a swab, we culture the bacteria, you test different drugs, and you test which one is working the best, and you give that to the patient. What if we could actually kind of, grow these tumors on a chip in defined matrices? So for example, a high-grade tumor would have exactly these composition of proteins, and you grow the tumors in these. You're capturing the heterogeneity in terms of stroma, in terms of the cells, and you basically add different drugs, you see which one is working best, and then you prescribe it to the patients. So we started thinking about coming up with a platform like that. And this is how it looks like. So we kind of, coated the wells, and this is a work that we did in collaboration with Ali. We coated the wells with matrix, had thin sections of the tumor, and we cultured them in defined media. Now, what is interesting is, if you don't actually have any matrix, so if you take the patient biopsy out, you cut it into thin sections, grow it on plastic, within 48 hours, you can actually see, this was at the time of, when you did the cultures, Ki67 is a biomarker for cell proliferation. Say 100% of the cells were proliferating. Within 48 hours, it drops to 25% of that. Okay. But when you have the right matrix, tumor matrix proteins, matched to the grade of tumor, you can see 75% of the tumors are still proliferating. On the other hand, if you have a low-grade tumor and a high-grade matrix protein, so you have changed the ECM, you can see it has dropped. So from here to here. So the cells are not proliferating as well. So there are cues being provided by the matrix that maintains that explant culture in the right state. So that's one piece of the puzzle. The second piece of the puzzle is basically that every patient has a completely different mix of growth factors and their receptors. And you can actually see this, these are patient data. You can actually see that they have completely different levels of growth factors and activation levels of those. So how do you activate those? Now remember, the tumor is being bathed by serum of the patient. So what we did was, in addition to the ECM, we also supplemented the tumor sections with serum of the same patient. And what happens is, when you do that, you can actually see autologous serum, the level of proliferation basically starts going up, even in the presence of versus EGF, for example, or just a control. So the signaling is affected by the addition of the serum. So what we have now developed is this platform system where we can actually screen for drugs that would actually be effective. And this is what we did. This is just validation that, when you combine serum and matrix protein, that's when you basically start mimicking exactly how it was, the tumor was when you started off. And in absence of that, you can see the profile of the tumor has completely shifted. This is basically the efficacy profiles, like, the phenotypic readouts. This is genetic transcriptome readouts. So if you don't actually maintain these conditions, you can see the transcriptome signatures have come. This is the original starting point. It is completely shifted if you don't have the right conditions, versus when you basically have these conditions, you start mimicking the original state. So now that we have the system, we started testing different drug combinations, okay, different treatment regimens. And we measured different parameters, like, for example, viability, histology, proliferation, apoptosis, multiple different parameters. And these came from 109 patients. So what we had created was, these explant cultures from these 109 patients. Each explant culture was exposed to a different drug. The patient also got one of those drugs. Okay. So we basically knew from the clinics that whether the patient is actually responding. So this example, this patient had a tumor here, and you can see after treatment, the tumor has shrunk. This was a partial response. The tumor was here, the tumor is still there, but it has shrunk partially. Here was a non-responder. That's a tumor, and you can see the tumor is actually now spreading and it's large. So we knew what treatments these patients have got. We knew the readouts of each of these treatments on each of these parameters. And so we built a machine learning algorithm to learn from these and assign scores of what each of these, this was an unbiased, analysis. So a score above 60 predicted a complete response. A score of between 19 to 60 was a partial response. And anything below 20 was basically a non-responder. So once we had established this training set, learned this training from this training set, we tested it out in 55 patients. And this is a subset of that. So remember, I talked about if you have wild-type KRAS patients, you will get a drug called cetuximab. So we had 13 patients in this pool who had wild-type KRAS. And based on that, all of these basically had the biomarker and got cetuximab. Only one patient responded. So remember, the target was present, the genetic target was present, but the response was one complete response, 2 partial response, and 10 non-responders. These patients got the drug, despite the fact that that they would actually not respond. In 7 days after this, post-diagnosis, based on our platform, we had predicted 2 of these patients would have complete response, 2 would have partial response, and 9 of these would have not responded. So this is a platform how we can actually kind of use machine learning and engineered explant cultures to predict who's going to respond and who's not going to respond. Now, these patients could have actually responded better to some other drug. and could have potentially been given that treatment. So this is where we are trying to kind of innovate. So this is a paper that was published in Nature Com, and this is the company that commercialized this platform. So now that we had actually built this, this explant culture platforms, this allowed us to ask fundamental questions. Why is it that the tumor still progresses? And I'm a pharmacologist, as I said. For me, this is a beautiful dose response curved. So we took cancer cells, and this is something that you might have seen in your experiments. So you have, when you expose these cancer cells to different toxic drugs, you can see that the viability is lost. And most people would say this is a nice dose response curve, including me. But what is interesting is that it never reaches zero. This is a pretty high, 1 micromolar concentration is a very high concentration. And at this stage, normal cells start dying. So why is it that it never reaches zero? Now, these cancer cells have never been exposed to these drugs before. There is no mutation to confer resistance against them. So how do they survive? Okay, so there is no mutational gain or inherited resistance that is coming up. So to address these questions, there are at that point of time, this theory was there that these cells are cancer stem cells. And these cancer stem cells cannot be killed by current chemotherapies that we use. Okay. And this is like, for example, Bob Weinberg is considered to be the champion of the cancer stem cell theory. Now, what is interesting is if you do an analysis of the number of cancer stem cells that actually is present, and this is a very nice study that was done, it's actually very, very low. And if you basically, in a tumor, and if you apply mathematical modeling on this, fraction of cells in a tumor, it doesn't add up in terms of the time to relapse. So if you have 0.046% of the tumors basically being a stem-like cell, for it to relapse, it cannot happen within the time frame in which relapse happens. So this paper, for example, basically kind of addressed this gap that existed. So how do these cells? What are these cells then? Are these all stem cells? These are definitely not all stem cells. So what are these? So we started asking this question, and what we did was these are the explant cultures from patients, and we treated. So here, for example, this is a vehicle-treated tumor. The same tumor, when we treated with taxanes, this is an old drug. This is the first drug of choice for breast cancer. This is breast cancer. And you can see what goes up is CD44. Okay. And CD44 is a biomarker for stemness. So what's happening is these didn't exist earlier. So each of these numbers are patients. And you can see with treatment, all these patients are basically, the tumors are now expressing higher levels of CD44. They're also expressing higher levels of CD24. Now, CD24 is supposed to be low in classical stem cells, but this also starts going up. And if CD44 goes up, so in this case, for example, here, this was breast cancer, treatment with docetaxel or doxorubicin, you can see CD44 levels are going up, but cleaved caspase is still low. On the other hand, this combination of drugs, carboplatin and gemcitabine, did not increase CD44 levels in the same tumor. This is from the same patient, but you can actually see cleaved caspase is going in. The cells are dying here. So effectively, if CD44 goes up, if the stemness marker goes up, these tumors do not respond to the existing drugs. Okay. So what we have been able to, what this suggests is basically there is a subset, it's not that, the stem cells exist per se, they exist, but there is also a tendency of normal cancer cells to transition to a stem-like state. And when that happens, we call them the drug tolerant states. So you take these cells, you treat them with the drug, there are still some cells which are surviving. And, this period is at very high concentration. And when you take the chemotherapy pressure off, and we waited for 30 days, and we basically then analyzed it again, these cells, which were not resistant, which were resistant at this point of time, became resistant again. So they were basically transitioning between two states, a stem-like state and a normal state. Normal state, they would respond to this. Stem-like state, they would stop responding. And the drug was inducing this. So we built a mathematical model based on this. And what we found is, in normal state, CD44 low cancer cells, this is the kind of cycle that happens. The moment you basically give chemotherapy, these CD44 low cells transition to a CD44 high, CD24 high state. We call it the hybrid state. And this hybrid state can actually move to a stem-like state too, but this state and this state do not respond to classical chemotherapy. So how do you then treat these tumors? What we found is when the cells move to this hybrid state, they activate a signaling pathway. So you can see CD44 is elevated compared to here. And this signal called phospho-HIC, it's a SRC family kinase protein, is also going up. And when you take the chemotherapy pressure off, after a few days, it goes back to normal. So at this stage, you can't kill the tumor with classical drugs. But what if you basically give a SRC family kinase inhibitor? And that's exactly what we did. So we had two schedules. You give taxanes first, followed by dasatinib, or you give dasatinib here at this stage when the signal is not present. And what we found is when you give this schedule, you basically get a survival advantage. Versus when you give this schedule, when the cells have reverted back to original state, dasatinib has absolutely no effect, which is here. Okay, schedule 2. If you give the two drugs together, there's a subtle improvement in survival, but not good enough. So scheduling becomes very, very important. You have to give, the temporal sequential administration of two drugs is important. So how does it translate into patients? So this is basically a biopsy from an explant culture from a patient. And what you see is that this patient basically had liver mets. And you basically see that when you treat, this is breast cancer, vehicle, you treat with taxane, very few dead cells. But when you give taxane followed by dasatinib, you can see that the number of dead cells are increasing. And this patient basically did this study. So what we propose is basically that, a parental cell, when you give chemotherapy, it moves to a state where all these CD24 and CD44 basically co-localize in a lipid raft, activate phospho-HIC, and that then gives a survival advantage. You take the chemotherapy pressure off, it switches back to this original state. And this CD44 high state is stem-like. So we're talking about we're talking about plasticity of cancer cells here, and the ability of taking one drug, killing some cells, but pushing the cells to another state and giving a second drug, which is only active in this state. So these are the kind of concepts that we can now introduce because we've engineered the right platform to study biology. And so this paper was published in Nature Com, and we followed up with the further characterization of these cells in terms of metabolic states. And we found that, the state basically also confers cross-resistance. So you give a taxane, the cells are also becoming resistant to doxorubicin, and why is that? So these were the two papers that were published. So this is how the tumor basically escapes. It starts switching its states. It starts, expressing different proteins to survive. And we have to tease out each of these pathways and shut them down. So I'll move to the next story, which is about immune evasion. And this is the current hot aspect. So why is it that the cancer cells can survive immunity? Now, our immune system is geared to basically clear out any cell that was not present during our childhood. But then cancer evolves later. So why is it that it can escape? And the reason that it can do that, it starts expressing all these proteins in high quantities. These are called immune checkpoints. And so what you have heard of very commonly are these two, PD-L1 and PD-1 interactions, or CTLA-4. If these immune checkpoints are elevated by the cancer cells, the T cells will actually start dying or become anergic. And Tasuku Honjo discovered PD-1 and PD-L1, and Jim Allison basically discovered CTLA-4, and they got the Nobel Prize for this. But the interesting thing is there are so many other immune checkpoints that can actually be targeted in addition to those two. So right now, we are still here. So it just kind of summarizes how smart a cancer cell is and how it can actually avoid or escape. How do we address these escape mechanisms? So why is it that so the fact that we have been so focused on these two also summarizes the results that we get. And in fact, what we typically see is only a subset of patients actually show this durable response. Maybe about 20 to 30% of the patients show durable response. Many of the patients basically have primary resistance. So 70% of the patients don't even respond to these existing immune checkpoint inhibitors. And some of them acquire resistance. So the fundamental questions to ask is, why does this happen? How does primary resistance evolve? What is acquired resistance to immunotherapy? These are the questions that, if we address, would allow us to come up with better therapeutics. And the second aspect is, so why is it so limited in terms of the durable response being only 20%? And how do we do that? The fact is, this is captured by this image. So this is how the immune system works. So effectively, you have the cancer cell. There are different mechanisms of immunity within the cancer cell, which are suppressed, needs to be activated. And these include sting, these include endogenous retroviral sequences, interferon responses. All this can potentially be activated. Now, once that happens, or in a normal immune response to a new cell or a alien cell or foreign cell would be that you would have macrophages getting triggered, you would have NKT cells getting triggered, you would have dendritic cells. This is what is called the innate immunity. Okay. And then you have adaptive immunity, which steps in after these cells have done this. The first layer of defense, the second layer of defenses is through the T cells, the B cells, the CAR T cells. These are all innate immunity. Where we are is right here, pretty much. We're playing only with these two cell types and a little bit of macrophages. NKT cells, B cells, dendritic cells, these are all frontier signs which needs to be addressed. The second aspect is all these cells will get triggered only if immunogenicity exists, if the cancer cell is highly immunogenic. So can we actually increase immunogenicity is a fundamental question to address. And that's by reducing its immunogenicity is a mechanism by which cancer cells can escape current treatments. And of course, as I said, we have to recruit all these cell types and not just T cells into the tumor for efficacy. So we started asking this question about, why is it that the cancer cells can actually avoid immune cells? And as I said at the start of my talk, that we address these questions using tools from nanotechnology. So we can actually look at these interactions at a scale what most labs can't achieve. So this, for example, is a cancer cell. And when we cultured them with immune cells, what we found is that the cancer cell sends out these very fine projections called nanotubes and connects with the immune cell. So here is another cancer cell. You can see the immune cells, which are rounded, much smaller in size. And you can see a long tube-like structure wrapping around this. I'll now zoom into this. So this is zoomed in here. These are the two immune cells. And you can see the tube-like structure here is now wrapping around this immune cell. If I zoom into this further at that magnification, you can actually see this tube. It's branching out and fusing with the immune cell. And what we found is that, so we label the mitochondria of the immune cell with a dye. So that's the green color that you see here. And we also label DNA with DAPI, which is blue in color. So when you merge green and blue, it looks purple. So this is a merge image, and you can see the purple color here, or the magenta color. That basically shows the mitochondria, because the mitochondria is the only other organelle other than the nucleus where DNA is present. You can actually see the mitochondria being trafficked on these tubes. So what's happening is the cancer cells sends out these projections, and mitochondria is the powerhouse, it's the battery of any cell. And T cells, for example, are exquisitely dependent on mitochondria for proliferation, for memory, for normal functioning. What it does is it basically, through these tubes, sucks away the mitochondria of the T cells, so that the T cells basically start dying. So what is interesting, I'll digress a little bit. This is something that we had actually seen in a previous study, not mitochondrial trafficking, but these tube-like structures. So what we had seen was, we were trying to replicate, we were trying to grow tumor embryonic bodies. But every time that we would grow an embryonic body close to a blood vessel, and the red is the blood vessel, these cells, instead of forming a embryonic body or a memosphere, would invade into the blood vessel. You can actually see the yellow cell inside the blood vessel. So we started thinking, we started looking at this at a very high resolution, and this is how it looks like. You can actually see the blood vessel here. That's the blood vessel. You can see the cancer cells, instead of forming these these ball-like structures, are aligning and spreading out on the blood vessel. And it sends out these nanotubes, creates a hole, and through which it can then metastasize. So this is how, applying the tools from nanotechnology allows us to study biology at these kind of resolutions. And what we found at that point of time is that through these tubes, we'd label this cancer cell with a dye, which labels all the proteins. And here you can actually see the tube. And this is an endothelial cell. It's injecting proteins through these tubes into the endothelial cell. So it's like a needle. It's actually kind of, it sends it out and transforms stromal cells around it. Okay. And this is what happens. So the cancer cells, so we did a culture where there was a cancer cell, and this is the tube-like structure. We basically found that, say, endothelial cell 1 received the signal, the next endothelial cell didn't. And we sorted them out into two different pools, these endothelial cells. The ones that received, that's the microRNA profile of the cell that received the signal. And that was the cell which was right adjacent to it and did not receive the signals, completely different microRNA profiles. And what happens is the cells that basically get injected with the microRNA, with the signal from the cancer cells, they actually become more sticky. Now think of it this way: the cancer cells are floating through the blood vessels. They dock onto an endothelial cell, which is lining the blood vessel, sends out these fine nanotubes, injects microRNA, and changes the cells, the endothelial cells, so that they become sticky. So that the next cancer cell that comes by, and these are the things, for example, that are actually getting altered. So CD137, it's a marker of stickiness of endothelial cells. So the next cancer cell can come and stick to that endothelial cell and then migrate out. And that's how you basically promote metastasis. So this was a paper that we had published in HCOM, and Jamisha did an amazing study on these cells. Now, coming back to what are these tubes doing in communicating between cancer cells and immune cells? So what we did was we labeled the mitochondria of the immune cells with a dye called mitotracker. And then we added it to cancer cells, which did not have the dye to start with. Okay, and these dyes don't actually leak out. So you can actually see a tube-like structure form. And in this case, you can see these two mitochondria on the tube. The same thing happens, as I said, we have explant cultures from patients. We labeled the immune cells from the patient with this dye, and then cultured with cancer cells which did not have the dye to start with. The dye is green in color, which is labeling mitochondria. And you can see the nanotube form between the immune cells and the cancer cells. And within about 16 hours, you can see the green mitochondria of the immune cells now present in the cancer cell. Okay. So what the cancer cells are doing are basically sucking away the mitochondria from the immune cells. This was just imaging, the same thing quantified using flow cytometry. So you can see in this case, we cultured the immune cells and the cancer cells together. So when we start, so the immune cells are labeled, the mitochondria is labeled with a green protein called dendra2. You can see at the start of the culture, these are the cancer cells, these are the mitochondria, these are the green mitochondria, positive immune cells, two different quadrants of the flow. And after 16 hours, you can see a double positive emerge. Okay, so these are cancer cells that now have gained mitochondria from the immune cells. But if I basically separate them out, they're no longer in touch with each other. You can see the two pools are still separate. Now, how do I know that it's unidirectional? How is it that the cancer cells are gaining mitochondria from the immune cells, not the other way around? So if you remember, I said the immune cells are smaller in size than the cancer cells. So we can actually kind of separate them out based on size. And you can actually see that these are the smaller CD3 positive immune cells. The red ones are the the larger sized cancer cells, and the blue are the double positive. And this double positive, the size is all aligned with the larger cancer cells. So what is happening is the cancer cells are sucking away the green mitochondria and now are becoming double positive. So this shows you directionality of trafficking. So what happens is, as the mitochondria, as the cancer cells are basically gaining mitochondria, does its oxidative phosphorylation go up? And so this basically, you can actually measure that using a technique called seahorse analysis. So this is your baseline reading. This is cancer cells when cultured alone. But when you culture them in conditions where they can hijack the mitochondria from immune cells, you can see the baseline, mitochondrial state or oxidative phosphorylation shoots up from here to here. So these are different parameters that you can actually measure. So this is basal respiration has shot up because of the cancer cells hijacking mitochondria. Their respiratory capacity has gone off, gone up, and that's also a sign of gain in mitochondria. And what happens is, as the cancer cells basically start gaining mitochondria or capability to do oxidative phosphorylation, they actually start proliferating more. And the immune cells, which have now lost mitochondria, so this is baseline immune cells, and this is when you co-culture, they've reduced in ability to do respiration. You can see spare respiratory capacity has gone down. Okay, those cells start dying over time. So then the question comes as to, how do you basically, does this happen in vivo? And what we basically did was we injected lung cancer cells into a mouse, which is genetically engineered to express green dye or dendra-2 dye in the mitochondria. And you can see the purple colors, these are the cancer cells. And you can actually see the green dots in the cancer cells. That's the mitochondria that has been gained from stromal cells by the cancer cells. So they're actually or the infiltrating immune cells. So the new hypothesis that we're actually now proposing is that in addition to basically shutting down the immune checkpoints, so say for example, these are the immune checkpoints, they would interact and you can shut them down using a immune checkpoint inhibitor. The cancer cells, what they do is, in addition to binding on immune checkpoints, they also send out these projections, they hijack the mitochondria out. And despite the immune checkpoint inhibition, these immune cells are inactive. They cannot actually kill the cancer cells. So what you now have to do is figure out how these tubes are formed and shut them down. And this is what we have done. We have started looking at different drugs that can actually perturb the process. We identified a drug that mediates that, the nanotube formation, a target. And I can see here is basically where the immune checkpoint inhibitor alone is in terms of inhibition of tumor growth. And when you combine with this drug, you basically start seeing inhibition of the tumor growth, correlating with increased T cell penetration and activated T cell presence in the tumor. So this is something that we've recently published in Nature Nanotechnology, and it has actually attracted a lot of attention. And so what is the next generation therapy? So based on this, this fundamental knowledge that we've gained using nanotechnology, now we have so this is the applied side of the story. What we found is that the tubes are formed by a protein complex called the exocyst complex. So here, for example, this is how it looks like. And you can see the red proteins, that's the exocyst complex. This is the cancer cell, that's the immune cell, that's the nanotube. You can see the red proteins are all localized on the cancer cell side of it, which is why the tubes are coming out, connecting with the immune cells. And if you shut down parts of these proteins using a siRNA knockdown, for example, you can actually inhibit mitochondrial transfer. So based on this, we have now started designing drugs that perturb this process. So we do structure-activity relationships. And so these graphs, for example, show that if these proteins are overexpressed, red, for example, that's a survival impact. So overexpression of these proteins means that less of the patients would survive over time with cancer. Whereas if these proteins are low, these patients are actually going to survive more. So the question is, can we actually shut down this pathway or degrade these proteins so that we can actually improve survival from here to here? And these are the drugs that we have now started designing. And you can see when you combine with immune checkpoint versus the immune checkpoint alone, this is the kind of inhibition that we see from here to here. So I'll summarize with, with this image. As I said, we have just figured out one new mechanism by which cancer cells can escape immunotherapy. But then the fact is we are still here. We are still activating the immune system. We have just started moving into innate immunity or activating macrophages, which I call the second generation immunotherapies. We are still a long way to go. For example, I envision the third generation immunotherapies would be B cell activators. There are recent papers that have shown that, if you have B cells in the tumor, you will basically have improved survival. And how does B cells work? They basically release IgMs. And you would have heard a lot about B cells from COVID. How different is treating COVID to basically treating cancer? It's the same immune response that needs to be triggered. So they release IgMs, and if that happens, they would recruit macrophages, they would recruit NK cells, they would recruit complements. They also basically act as antigen-presenting cells to T cells. So the question is, could we activate these next second or third generation immunotherapies? That's the frontier science. So what we have been able to do, or and I think this is the community that hopefully I'm addressing today. This poses a lot of questions that, could we build tools from engineering to address, these unmet needs? Could we learn from other areas of immunology, virology, for example, which can impact treatments? And I just wanted to summarize that if we can actually use the right tools, for example, from nanoscience, I think we can actually address a lot of fundamental biology. There's a lot to be explored. And with that, I would, thank Mehmet, Ali for hosting me and these funding agencies for supporting this work. Thank you so much.
Thank you for the fascinating seminar. I appreciate it. A lot of questions came in, so let me read the questions. The drug you use for anti-angiogenesis around the tumor cells does not interrupt other organs' healing process.
Yeah, so yes, theoretically, anti-angiogenesis drugs, angiogenesis is very, very important for healing, right? For wound healing. But what happens is when you basically have a cancer patient, it's a question of time, right? You have to basically increase the lifespan of a patient. And once that happens, you can deal with impaired wound healing later. But most cancer drugs would actually impair wound healing, by the way. Okay, it's not just anti-angiogenesis drug. So it's a question of prioritizing. And the priority when you have a cancer patient is to improve survival of the patient.
Thank you. Does the breast cancer stem cells express ALDH1, ALDH3, or other cancer stem cell markers?
Yes, they do. Yeah, so there are other cancer stem cell markers that are actually and we have seen some of them. They're actually even the hybrid cells that we get express some of those biomarkers.
Another question came in. What type of T cells do you use? Is it primary isolated or cell line of T cells?
That's a good question. So for screening, we do actually use a cell line, and we don't need it to be activated. The immune cells, the cancer cells, for some reason, basically can detect immune cells and hijack the mitochondria. And we do use primary. We are also using activated T cells.
Thank you. Does the mitochondria extraction is by cancer stem cells or all cancer cells?
Great question. We don't know. So what is interesting is it's only 50% of the cancer cells which were positive for mitochondria from the immune cells. Okay, so there's a subset of cancer cells that basically gain the mitochondria. There's a subset of cancer cells that did not. So there may be, and in fact, that's one of the questions that we're exploring, that it could be that, there is a subset of cancer cells, maybe the stem cells, which are actually more active in hijacking mitochondria. So we are studying the cancer cell heterogeneity in terms of its capability to harvest mitochondria.
Thank you. Can cancer cells extract the mitochondria from CD4?
Yes, they can. They can also extract mitochondria from macrophages, for example. So we are now starting to expand the pool of cells from which it can actually hijack mitochondria.
Okay. What about the activated CD8 T cells therapy on cancer cells?
Yeah. So just randomly activating T cells is not going to be activated. T cells is not going to kill a cancer cell. You have to have the antigen being recognized, right? And that's what you do with CAR T cells. So one of the studies that we're doing, we basically are taking T cells that are activated against an antigen that is expressed by the cancer cells. So it's very important to have the right antigen and the cancer T cells that are expressed by the cancer cells and the T cells that are activated against those antigens.
Thank you. This one is curious about the PD-1 and PD-L1 gene silencing. Is it better for therapy?
No. So if you can actually kind of if you can silence it in on the cancer cells, it's the same effect as basically in the as using an immune checkpoint inhibitor, right? So that's yes. I mean, it would be like using another immune checkpoint inhibitor. The question is, can you actually silence it effectively? I don't know.
See. In place of immunotherapy, can we target mitochondrial pathway to inhibit cancer?
Yes, people are trying to do that. And there is a lot of metabolic dysfunction is a hallmark of cancer, and being able to stabilize that and shut down mitochondria of cancer cells, absolutely, you can potentially have very powerful effects.
Thank you. What is the contribution of the immune system in driving evolution, genetic mutations of cancer cells? Do you think that HLA polymerism is a contributor, or if so, could it be used to evaluate the mutagenic potential of cancer cells?
Okay. So look, I mean, so if basically to answer this question, I think we need to understand that maybe there is a different levels of immunogenicity. So the tumors, the cells that are highly immunogenic are going to be eradicated. So the clones that are less immunogenic will start evolving, and you will start selecting for cells that are less immunogenic. And then the question comes, then you need treatments or therapeutic strategies that can increase the immunogenicity of those left cells. Otherwise, they will evolve and they have beaten the immune system.
Can we consider the extracellular vesicles, such as exomer, as cargoes of RNA to find a novel solution for cancer treatment?
Yeah. So there are people who are trying to kind of work on that. For example, cancer cells do release exosomes that can actually reprogram in the stromal cells, and shutting down exosomes can potentially prime the immune cells as well as the stromal cells to suppress tumor progression. Yes, that's a very hot area of research right now.
Thank you. Do those resistant cancer stem cells present a highly proliferative phenotype? If not, would it be one of the reasons for chemotherapy not to be efficient?
Okay, so in fact, one of the most resistant cells are actually not proliferating that much. So if a tumor doesn't proliferate as much, it is actually hard to treat. It's the other way around. It's not that, cancer cells that are proliferating very fast probably are the most sensitive to treatment. So it's opposite of what the question is.
I see. Thank you. In your opinion, macrophage recruitment to a tumor will increase T cell presence as well?
Yes, could actually, because the thing is that, Macrophage basically is an antigen-presenting cell, and they also release chemokines and cytokines, which would actually kind of prime immune response. So the whole idea is to have both adaptive and innate immunity kind of work together in tandem. So you might actually need to basically activate both for a proper response. See, the problem that happens is, we have everybody jumped into T cells because that's the main killer, right? But innate immunity is also a very important piece. And in our immune system, it's not just, the T cells. You actually have all the cells working in tandem. So to really have an impact, you have to look at the forest, not just the tree.
Yeah. Well, that's all the questions I have. Thank you for your time, Shiladitya. Fully appreciate it. There was so many questions.
My pleasure, Mehmet.
Thank you.
Thank you so much. See ya. Bye.
Have a nice day.